eventually get a high enough N that it comes down to a small amount. If you repeat it enough, you'd be surprised. It actually comes right on top of theory, and it's really fantastic. So sort of in summary of that project, very gradual, like science. So it was sort of in summary of that project is I was more heartened that we were thinking about aging in this sort of correct way, that there was this energetic trade-off between an organism's early life and its late life, and that could explain a lot of how aging works. That's sort of a very general thing to say, and eventually we want to come to what molecules are directly regulating aging, and that's sort of what we'll talk about for the rest of this. And one of the things we want to go after were sirtuins, and it's been sort of the bane of the aging field for a long time. And one of the research projects we generated was based off of trying to fight how people were analyzing this paper. So if people sirtuins were a molecule that were purported to be downstream of dietary restriction, and dietary restriction we knew extends lifespan in so many different cases. And one of the forms of evidence that came out for that was if you knock out sirtuins in mice, this homozygous knockout, and you do dietary restriction on top of that, you don't get a lifespan extension anymore. Whereas in wild type, you still get this lifespan extension. And we've shown in yeast that this mode of thought just doesn't work, and we wanted to provide more evidence for why exactly that might happen. And I kind of want to model this section of the talk based off, I don't know why I keep bringing Richard Miller up, because he says some pretty cool things. I went to a talk 2 years ago, and he just showed 2 slides. One, he just said sirtuins don't have anything to do with anything. Resveratrol doesn't affect lifespan at all, so this is a putative activator of sirtuins. So I called everybody, trying to make sure everything was working right. Didn't, a ton of people put a lot of time and effort into this, nothing worked. He was very frustrated by it, obviously. And then he went on, hey, this works, we should really be studying this, the reduction in translation sort of mechanism of aging. And that was really cool, and I hope I'll sort of follow that sort of thought in this presentation here. And so George Sliffen and I spent about 1.5 years creating all these long-lived mutants in a SIR2 deletion background strain. So SIR2 deletion mutants are very short-lived. They sort of have the same effect as you see in mice. They're much shorter-lived than wild-type yeast. And we thought that, surely something that's long-lived should be able to fix this effect, right? Well, we tried 30 different cases, and in every single case, not a single one of these long-lived mutants was able to sort of extend the lifespan of a SIR2 knockout. And you can interpret that to say that sirtuins are downstream of every single longevity pathway that we've ever tried in these. Or you can say that when an organism is short-lived, it's not necessarily informative in terms of adding something on top of that. You can't tell if it's downstream of that longevity intervention. And the additional proof that we had for that sort of way of thinking about it is it's been known for a while that you can remove sort of a yeast-specific form of DNA damage, this ERK formation, by deleting this protein FOB1. And this is the only thing that we know extends lifespan that can rescue SIR2 mutant. So if we delete FOB1 in the SIR2, it goes back to about wild-type levels. It doesn't have that DNA damage present in the cell anymore. And now what you see is you take these mutants, which are thought to be sort of within the dietary restriction pathway, and you can get a lifespan extension. So this is sort of the SIR2 FOB1 curves, which die off very quickly as they age. Whereas you put this longevity extending intervention in it, and you get a longer lifespan. So it was very clear that SIR2 was not required for lifespan extension in these cases. And I think it's a very important concept to carry on to whatever you're doing is if you're using something that's very sick, is it going to be an informative background to use in your assays? And for me, it boils down to one thing, which I've shown in a couple talks before, is the sort of hammer hypothesis. If you have something whacking your long-lived yeast with a hammer, in this case a SIR2 deletion, it's going to injure the cell. But that injury has nothing to do with aging whatsoever, right? So if you have something that's extending lifespan, it's not going to be able to overcome this effect of something that's really damaging to the yeast that has nothing to do with longevity. That being said, somebody got very rich off of this concept that sirtuins were downstream of dietary restriction. He sold his company for $720 million based off of what I believe is a sort of false concept. And so hopefully if we figure out something correct for this mechanism of dietary restriction, we might be able to get a lot more funding than $720 million. But time will tell. Yeah. So George and I were both frustrated with sirtuins, and we went off after some different things. And the story, which will follow the rest of the presentation, came off of Matt sent me to China. He was trying to outsource our work, and
maybe I was trying to outsource the graduates.
yeah, trying to get me out of the lab because I was annoying him too much. And what came out of that was after Matt left and people were sort of more relaxed and able to talk to me personally, and we got to know each other. They sort of very humbly came up to me and said, you know, it's great that you're teaching us this replicative lifespan assay. It's a really cool assay, but honestly, we don't know how to do science. So can you teach us how to do science? And I was a second-year graduate student, and I was like, why is somebody asking me how to do science? But you know, being who I am, I didn't just say, I don't know. I said, I'll try to help you out and create a presentation and do what I envision to be how you go about a research project. And sort of following what they usually do, they picked a random gene and went after it. And the random gene that I picked was from our long-lived screen. I only had one night, so I didn't have much basis to go off of. But since they were sort of looking at this gene, I decided to show them how they could build a project off of that. And LOS1 in particular is something that caught my eye as being something that we could create a story out of really quickly. And it's because it's a conserved nuclear tRNA exporter. And just looking at that description in a database, I could already sort of plan out how this mutation was extending lifespan. But I went into sort of the background research to try and explain how you go about things. And I'll recommend to anyone the first basis of going after any project is you read a lot of review papers. And a lot of us already know that dietary restriction is the best known method for extending lifespan in a wide variety of cases. And people will still argue about that to no end, but it's really cool that it extends lifespan in so many model organisms: yeast, worm, flies, mice, most recently even in monkeys. And I think a lot of people in the aging field are sort of sick of seeing this slide, but when I first saw this slide, I just broke out in laughter, and I couldn't it wasn't because this guy looked a little bit angry, even though he was healthy. It was because I saw the axis on this graph, and I was like, who has this on their axis in 30 years? Who does an experiment for 30 years? So somebody was very interested in this concept, and I'm glad somebody felt it was a really important study to show. And they really did show that dietary restriction is a really wonderful intervention. And so, kind of background to the LOS1 project, I thought, okay, this is something that might fit into the dietary restriction pathway. And here's why. So if nuclear tRNA, or this nuclear tRNA exporter, LOS1, doesn't function anymore, so we delete it, so we get a buildup of nuclear tRNA, how might that affect the cell? It's sort of obvious, right? We've always thought of tRNA as something that just goes to ribosomes and builds proteins, right? So if you don't have enough tRNA going to the cytoplasm, you're not going to get as much protein. And we already knew that you can extend lifespan in both C. elegans and yeast by reducing translations. So this was a yeast paper done by Kristen Steffen in the Kennedy lab a few years ago, and also from Liz Kenyon's lab in 2007, where they, in both cases, reduced the rates of translation and found a wide variety of mutants that could extend lifespan. So it seemed like, okay, if you get tRNA restricted to the nucleus, surely you get a reduction in translation. That's why it extends lifespan. And there was another reason I thought it would be in the dietary restriction pathway, doing further background research, who actually researches tRNA and its And the Hopper lab did a lot of work on this, and she found out that if you deprive yeast of various forms of nutrients, different forms of dietary restriction, you can get the buildup of tRNA in the nucleus of these cells. And so this was sort of heartening to me that it could be something that's regulating dietary restriction. And so I decided to just go ahead and test that. So we've tested the Losman deletion so many times, it's very clear that it's long-lived compared to wild-type yeast. And what was sort of new is that we could put this Losman deletion in that sort of background where you don't get that yeast-specific form of DNA damage, so 251 background. And you get this huge lifespan extension. And if you want to show this in the dietary restriction pathway, you try dietary restriction on top of that long-lived mutant and see if it additionally extends lifespan. And indeed, there was not a significant extension of lifespan when you combine those two interventions. So this is sort of promising to say that, okay, this mutant could be within the dietary restriction pathway. We're starting to build something out of this initial observation. And very importantly to me, it did not extend the SIR2 mutant, so it wasn't some sort of freak mutant that was like FOB1 and only yeast specific. That was very good to see. So we could alter lifespan by getting nuclear tRNA by getting rid of a tRNA exporter, right? Well, there's also something that's known to be involved in tRNA import, and that's this protein MTR10. And so this was shown that there's this sort of very special way tRNA are processed in yeast. So in most organisms, you just simply create the tRNA in the nucleus, go through all the processing events, and then it gets shipped out to the cytoplasm for translation. In yeast, it has this sort of altered route to its final maturation, where it gets synthesized in the nucleus, then it moves out to the cytoplasm for some reason and gets an intron removed, and then it goes back to the nucleus again. And that process of going back to the nucleus was regulated by this protein MTR10. And so if you did dietary restriction, you got to build up a tRNA in the nucleus again, but if you remove this protein, you no longer get that buildup. So it might be possible that the lifespan effects might work out the same way. So if you get rid of MTR10, you no longer get this retrograde transporter tRNA to the nucleus. And you also no longer get a lifespan extension here. So MTR10 completely blocked the lifespan extension by dietary restriction. And we sort of played around with ways to overexpress genes in yeast, and we found that if we overexpress this thing, which should favor nuclear tRNA, it also extends lifespan. So these were sort of two different methods for showing that, okay, increased amounts of nuclear tRNA could extend lifespan. That's great. But throughout all this, I was frustrated to the point of I thought I must be getting tons of suppressors in my yeast, tons of mutations. And that's because the Losman deletion, which we know builds up tRNA in the nucleus, has absolutely no effect on growth rate, no effect on ribosomal translation. I think that's a very important point. And we assayed this through, we sort of overkilled this just to make sure we were right. Traditionally, what you can do in yeast to see if there's sort of a background mutation to see if that's affecting things, you can cross it with a wild type cell. And then every other spore that comes out of meiosis should have this deletion in it. And sometimes you would see a small colony if there was a possibility that LOS1 deletion was slowing translation. We never saw that. Whereas in our control slow translation mutant, we saw, as we would expect, slower growth rate in cells. And if we just do strictly growth curves, we saw it overlap with wild type completely. So it wasn't even a subtle effect of reduced translation. We did ribosomes, how many ribosomes were bound to an mRNA at any given time, which is a direct measure of translation. And we found that, unlike our control case, which did reduce translation, LOS1 didn't have any change in the number of ribosomes bound to an mRNA. And similarly, if we do cell cycle, it looks exactly like wild type again. It doesn't have this G1 delay like most other long-lived mutants do. And this was incredible to me that we found something entirely novel and new about tRNA that I hadn't thought of before. Is that tRNA could actually be acting as a signaling molecule. And to me, that's a really incredible concept. People have always thought of it as something that's being directly involved in translation, but it seems like there are things that regulate where tRNA is in the cell. It can actually bind to factors and cause specific rates of transcription on certain genes. And And so I wanted to sort of build up a project where I would find out what exactly was this nuclear tRNA creating in the cell. What was its signal? And eventually I come up with this diagram where we go all the way from dietary restriction through a DNA damage sensing molecule, regulates LOS1, and it gets down to the downstream event, activates a transcription factor, which seems to be completely involved in longevity. And I'll just go through these one by one and show how we found that out. So the first step was I did a little bit more background research for what exactly could regulate LOS1. And I found that people discovered that either by reducing nutrients or hitting cells with a form of DNA damage alkylating agent, MMS, you could get this redistribution of LOS1, which is normally active in the nucleus, to other places in the cell, more diffuse throughout the cytoplasm. So these are terrible pictures, but they still published in this, what people are sort of expecting as a standard. LOS1 is generally nuclear under an unstressed condition, but if you add a stress of MMS, it becomes more diffuse. And this diffusion event was dependent on whether or not Rad53 was in the cell. So if you deleted Rad53, you still got a nuclear accumulation of this LOS1 protein. And me being naive for one, and wanting to just try a microscope of GFP proteins, that sounded pretty cool. I wanted to see if dietary restriction could have the same sort of effect. And just having a little bit of adventure, trial microscope, got some pretty cool results. So if we dietary restrict yeast, LOS1 normally is in the nucleus here, but it becomes more diffuse throughout the cytoplasm here under dietary restriction. And this relocalization event was completely dependent on this DNA damage sensing protein, Rad53. And that was interesting to me because it sort of implicated a DNA damage response pathway within the dietary restriction pathway. It was just the quantification of that. So you can reduce LOS1 quantity in the nucleus, probably get a tRNA buildup, and then you no longer get that if Rad53 is deleted. Now I want to know, okay, well, Rad53 was necessary for dietary restriction to have an effect on LOS1. Does dietary restriction itself have an effect on Rad53? That would more conclusively put it within this pathway. And it was interesting that when we looked at it on the microscope, we could see a little bit brighter cells in it, but traditionally what I've been doing was quantifying the amount in the nucleus where the DNA actually is. We saw a very modest increase of Rad53 compared to the cytoplasm in the nucleus, compared to our control DNA damage cell. But if we took these cells and ran them through a flow cytometer, something that can measure the amount of GFP in any given cell, on average we found that dietary restricted cells had about twice as much Rad53 GFP in them when the cell was dietary restricted. So this was promising in the sense that it showed that Rad53 was not only necessary for relocalizing LOS1 under dietary restriction, but was also modulated by dietary restriction on an expression basis. And of course, when we get all these preliminary data, we want to take them into lifespans and see what is the actual aging effect. And it's sort of as we predicted, if you delete Rad53, something that senses DNA damage, it's a very short-lived cell. It only divides about 6 times compared to wild types 25 times. But if you do dietary restriction on top of that, you don't get a lifespan extension. So that was one piece of evidence that was showing that Rad53 could be downstream of dietary restriction. Of course, I don't like that line of evidence because it's very similar to what happens in the SIR2 deletion, where it just shortens lifespan so you can't really find out anything. But what was sort of cool to find was that we deleted LOS1 in this, something that we thought was downstream of Rad53. And we got a rescue of lifespan, at least partially, so it could really extend from this Rad53 single deletion to the LOS1 Rad53 double deletion. And this was sort of indicative of something that could be downstream of Rad53. So it's really cool. We could put Rad53 between dietary restriction and LOS1. And similarly, if we overexpress Rad53 itself, in the system we got a modest lifespan extension. And so all that work was just to show these two coupled arrows here. And we wanted to include whether or not some TOR1, something that we knew a lot about in terms of aging, how that related to this pathway. And we thought it should be a lot like dietary restriction, where if we delete TOR1 in